Welcome back friends. Welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding mitosis and its regulation from cell cycle and cell division in detail. So here mitotic phase or M phase, this is the most dramatic period or this is the most dramatic phase of the cell cycle. During the mitotic phase in general cell division occurs. So here when in single parental cell, when it participates in mitotic cell division, it yields two daughter cells, and here these two daughter cells, they remain identical in case of size, shape and even in case of the chromosomal number. So the reason why mitotic cell division, or it is otherwise known as equational division. Is it clear? So let me write it on the board. Mitotic phase or M phase is the most dramatic period of the cell cycle during the mitotic phase in general cell division occurs, right? That means when in single parental cell, so let me show it here. When in single parental cell, when it participates in mitotic cell division, it yields two daughter cells. So here these are nothing but what? The daughter cells, right? So here the daughter cells which successfully synthesizes during the mitotic cell division, they remain identical in case of size, shape and even in case of the chromosomal number. That means if the specific parental cell, if it has 46 chromosomes, obviously both the daughter cells, they should possess or they should contain 46 chromosomes. So the reason why mitotic cell division, or it is otherwise known as equational division. Is it clear? So let me write it here. During the mitotic cell division, when in single parental cell, when it participates in mitotic division, it yields two daughter cells, right? And here these two daughter cells remain identical in size, shape and even in case of the chromosomal number, right? So the reason why mitotic cell division, or it is otherwise known as equation division. And here as a specific mitotic cell division, it is a progressive process. Here we cannot understand or here we cannot underline the accurate imaginary lines of the specific mitotic cell division. Is it clear? As the specific mitotic cell division, it is a progressive process. Here we cannot underline the accurate imaginary lines of the specific mitotic cell division. And however, in order to understand or here in order to study the specific mitotic cell division, here we can take the advantage of four different phases. And here these four different phases, they may include prophase, metaphase, anaphase and telophase. Is it clear? So here by taking the advantage of these four different phases, here easily we can study the accurate mitotic cell division in detail. So let me write it on the board. As the mitotic cell division it is an progressive process, right? Progressive process. That means the specific mitotic cell division, it successfully proceeds from one particular phase to another phase. So the reason why here we cannot underline the accurate imaginary lines of the specific mitotic cell division. As the specific mitotic cell division, it is in progressive process, we cannot underline the accurate imaginary lines of the mitotic division, mitotic cell division, right? And here, in order to understand the specific mitotic cell division, here we can take the advantage of 4 different phases. So here, by taking the advantage of 4 different phases, easily we can understand the specific mitotic cell division in detail. So here, by taking the advantage of 4 different phases, easily we can understand the mitotic cell division, right? The mitotic cell division. And here the 4 different phases, they may include prophase, metaphase, anaphase and telophase. So at first we will be studying regarding prophase. So here, what is prophase? So let me arrange the things here. So as already mentioned, the mitotic phase or M phase, it can be well studied or it can be well explained under 4 different phases. And here these 4 different phases, they may include prophase, metaphase, anaphase and telophase. So let me write it here. Mitotic phase can be well studied under 4 different phases, and these may include prophase, metaphase, anaphase and telophase, right? So at first we will be studying regarding prophase. So here prophase, it is the specific phase which generally occurs after the successful commencement of S phase and G2 phase. That means after the successful commencement of S phase and G2 phase of the interphase stage of the cell cycle, in general prophase comes into the picture. And here prophase, it is the first stage of the karyokinesis. As we all know, mitotic cell division in general, it begins with division of nucleus, which is otherwise known as karyokinesis, and it ends with division of cytoplasm termed as cytokinesis. So here, division of nucleus or karyokinesis in general, it begins with prophase. Is it clear? So let me write it here. Prophase, this is the first stage. This is the first stage of karyokinesis. So here, what exactly happens here is, during the S phase, as we all know, the parental DNA, it participates in replication in order to yield the daughter DNA. So here, both the parental DNA and even the daughter DNA, it remains in intertwined conformation during the interphase stage, because during the interphase stage, the specific chromatin material, it remains in decondensed form. Is it clear? So let me show it here. So let us assume it is a parental DNA, and here it is daughter DNA, right? Daughter DNA. So here both the parental DNA and even the daughter DNA, it remains in intertwined form. That means they won't remain in separate form during the interphase stage of the cell cycle, because during the interphase stage of the cell cycle, the specific chromatin material, it remains in decondensed form. So the reason why both the parental DNA and even the specific daughter DNA, they remain in intertwined form during the interphase stage. But here, once when the specific cell cycle, once when it successfully proceeds from the interphase stage to the mitotic phase, now here the specific chromatin material, it starts remaining in condensed form. Is it clear? If you look at here, during the early prophase stage, the specific chromatin material, initially which is under decondensed form during the interphase stage of the cell cycle, now here the specific chromatin material, it starts remaining in condensed form. So just look at here, chromatin material which is under condensed form. So here, once when the specific chromatin material, when it remains in condensed form, now here the specific chromatin material, here it is otherwise known as chromosome. Is it clear? So let me write it here. During S phase, the chromatin material remains in decondensed form. But here, once when the specific cell cycle, once when it successfully proceeds from the interphase stage to the mitotic phase, now here the specific chromatin material, initially which is under decondensed form, it successfully starts to convert into condensed form. Is it clear? And here the condensed form of the chromatin material, here it is otherwise known as chromosome. The condensation of the chromatin material starts during the early prophase, right? Early prophase. And here the condensed form of the chromatin material, here it is otherwise known as chromosome. The condensed form of the chromatin material is termed as chromosome, right? So here basically a specific chromosome, it has two sister chromatids. So if you look at here, easily we can understand. So let me show it here. Basically a specific chromosome, it has two sister chromatids. So here these are nothing but what? The sister chromatids, right? Sister chromatids. So here these sister chromatids, they are successfully aligned by taking the advantage of in single centromere. Is it clear? These two sister chromatids, they are successfully aligned by taking the advantage of a specific structure termed as centromere. So the reason why here these specific chromatids, they are termed as sister chromatids. Is it clear? And here in case of animal cells, here we can observe one more other unique feature in case of the prophase of the mitotic cell division. In case of animal cells, during the S phase, the specific centrosome which undergoes successful duplication. Is it clear? If you look at here, the specific centrosome which successfully undergoes duplication during the S phase in case of the animal cell. Now here the specific centrosome, it successfully migrates towards the opposite spindle poles. Is it clear? The specific centrosome which successfully undergoes duplication during the S phase of the cell cycle. Now here the specific centrosome, it successfully migrates towards the opposite spindle poles during the early prophase. Is it clear? So let me arrange the things here. So as already mentioned, in case of animal cells, the specific centrosome which undergoes duplication during the S phase of the interphase stage of the cell cycle. Now here these specific centrosome, they are ready to migrate towards the opposite spindle poles at the very beginning of the early prophase. Is it clear? So let me write it here. During the early prophase, the centrosome which undergoes duplication during S phase, ready to move towards the opposite spindle poles, right? And here, when the specific mitotic cell division, when it successfully progresses from the early prophase to the late prophase, now here at the very beginning of the late prophase, here we can observe the disintegration of the nuclear envelope. Is it clear? As we all know, each and every nucleus, it is protected by the means of a specific outer covering layer which is termed as nuclear envelope. At the very beginning of the late prophase, here we can observe the disintegration of the nuclear envelope. So here when the disintegration of the nuclear envelope, when it successfully takes place at the very beginning of the late prophase, now here the various subcellular organelles like endoplasmic reticulum, Golgi complex, nucleolus, all these specific subcellular organelles, they disappear at the very beginning of the late prophase. Is it clear? So let me write it here. At the late prophase, the disintegration of the nuclear envelope, occurs. Nuclear envelope occurs, right? The various subcellular organelles, like endoplasmic reticulum, Golgi complex, and even nucleolus disappear, right? So here this is regarding prophase, and then after we will be studying regarding metaphase. So here what is metaphase? So let me arrange the things here. So after the successful commencement of prophase, the next specific stage of the mitotic cell division, here it is nothing but metaphase. So here what are the various cell features which are most commonly observed during the metaphase stage of the mitotic cell division? So as already mentioned, at the very beginning of the late prophase, disintegration of the nuclear envelope takes place. And in addition to the disintegration of nuclear envelope, the various subcellular organelles, they also successfully disappear at the very beginning of the late prophase. So here, because of the disintegration of the nuclear envelope and the disappearance of various subcellular organelles, it is responsible for the successful spreading out of chromosomes throughout the cytoplasm of the biological cell. Is it clear? because of the disintegration of the nuclear envelope and the disappearance of various subcellular organelles, it is responsible for the successful spreading out of chromosomes throughout the cytoplasm of the biological cell. Is it clear? So let me write it here. Metaphase. During the metaphase stage, the chromosomes are widely spread out throughout the cytoplasm of the biological cell. Throughout the cytoplasm of the biological cell. Biological cell, right? And here, as already mentioned, the specific centrosome which generally duplicates during the S phase. Now here the specific centrosome, they are ready to migrate towards the opposite spindle poles during the prophase of the mitotic cell division. Now here the specific centrosome, they are capable to remain in association with the cytoplasm of the biological cell by taking the advantage of ester microtubules. Is it clear? The specific centrosome will successfully migrate towards the opposite spindle poles. These specific centrosomes, they are capable to remain in association with the cytoplasm of the biological cell by taking the advantage of ester microtubules or non-kinetochore microtubules. So here the specific centrosome in association with the ester microtubules and the specific chromosomes which are aligned by taking the advantage of spindle fibers, it is together termed as mitotic apparatus. Is it clear? And here the condensation of the chromosome, initially it begins during the prophase, and here the condensation of the chromosome, it successfully completes when the specific cell division, when it successfully reaches to the metaphase stage of the mitotic cell division. Is it clear? The condensation of the chromosome in general, it begins during the prophase, and here once when the specific cell division, once when it successfully progresses from the prophase stage to the metaphase stage, here the condensation of the chromosome, it successfully completes. Because of the successful completion of the condensation of the chromosome, now here we can visualize the accurate structure of the chromosome during the metaphase stage of the mitotic cell division. Is it clear? So let me write it here. Condensation of the chromosome. Condensation of chromosome completes during the metaphase stage. During the metaphase stage, right? And here chromosomes are visible under microscopic observations. Basically one specific chromosome, it has two sister chromatids, and here both the sister chromatids, they are aligned by taking the advantage of a specific structure termed as centromere. Is it clear? So let me show it here. So here these are nothing but the sister chromatids, right? And here it is nothing but the centromere. So here basically specific chromosome, it has two sister chromatids which are aligned by taking the advantage of one specific centromere. And here on the surface of the centromere, here we can observe a specific disc-shaped protein complex termed as kinetochore. Is it clear? On the surface of the centromere, here we can observe a specific disc-shaped protein complex termed as kinetochore. So here by taking the advantage of kinetochore protein complex, the specific chromosomes, they are capable to remain in association with spindle fibers. So let me show it here. So here it is nothing but kinetochore, right? Kinetochore protein complex. Kinetochore protein complex. So here the specific chromosomes, they are capable to remain in association with the spindle fibers by taking the advantage of a specific protein complex termed as kinetochore protein complex. Is it clear? And here the specific chromosomes, by taking the advantage of kinetochore protein complex, they are capable to remain in association with the spindle fibers. Now here, once when the specific chromosomes, once when they are capable to remain in association with the spindle fibers by taking the advantage of kinetochore protein complex, now here the specific chromosomes, they are successfully aligned at the equatorial region. Is it clear? So let me arrange the things here. So as already mentioned, when the chromosomes, when they remain in association with spindle fibers by taking the advantage of a specific protein complex termed as kinetochore. Is it clear? When the chromosomes, when they remain in association with the spindle fibers by taking the advantage of kinetochore protein complex, now here the chromosomes, they are ready to migrate towards the equatorial region. So here the specific region at which the chromosomes are ready to migrate towards the equatorial region, here it is termed as metaphase plate. Is it clear? The specific region at which the chromosomes are aligned at the equatorial region, here it is termed as metaphase plate. And here, as already mentioned, one specific chromosome, it has two sister chromatids, right? So here one of the specific sister chromatid in association with the centromere, it successfully exposes to one spindle pole. Is it clear? One sister chromatid in association with the centromere, it successfully exposes to one spindle pole, and the other sister chromatid in association with centromere, it successfully exposes towards the opposite spindle pole. Is it clear? So let me write it on the board. During the metaphase stage of the mitotic cell division, chromosomes are ready to migrate towards the equatorial region, and here it is otherwise termed as metaphase plate. Is it clear? Metaphase plate. So here, as we all know, one specific chromosome, it has two sister chromatids. One of the sister chromatid in association with centromere, it successfully exposes to one spindle pole, and the other sister chromatid in association with centromere, it successfully exposes towards the opposite spindle pole. Is it clear? Chromosomes which has two sister chromatids are exposed towards the opposite spindle poles. Is it clear? So here this is regarding metaphase stage of the mitotic cell division in detail, and then after we will be studying regarding anaphase stage. Is it clear? So let me arrange the things here. So when the mitotic cell division, when it successfully progresses from the metaphase stage to the anaphase stage, during the anaphase stage, splitting of the centromere takes place. Is it clear? During the anaphase stage, splitting of the centromere takes place. So here, because of the splitting of the centromere, now here the sister chromatids, they are ready to migrate towards the opposite spindle pole. Is it clear? So here, because of the splitting of the centromere, the sister chromatid, they are ready to migrate towards the opposite spindle pole. Initially, the specific parental cell, it has 46 chromosomes, but during the S phase, 46 chromosomes, they become 92 chromosomes. And out of the 92 chromosomes, 46 chromosomes, they successfully migrate towards one spindle pole, and the other 46 chromosomes, they successfully migrate towards the opposite spindle pole. Is it clear? So let me write it here. During the anaphase stage, splitting of centromere takes place. So here, because of the splitting of the centromere, the sister chromatids, they are ready to migrate towards the opposite spindle pole. Is it clear? Because of the splitting of centromere, sister chromatids are ready to move towards opposite spindle poles. And here, these sister chromatids will successfully migrate towards the opposite spindle pole. In future, they act as chromosomes in the daughter nuclei. Is it clear? These specific sister chromatids will successfully migrate towards the opposite spindle pole. These sister chromatids, they act as chromosomes in the upcoming daughter nuclei. Sister chromatids which migrate towards opposite spindle pole act as chromosomes in the upcoming daughter nuclei. Isn't here? We need to understand one of the key points which takes place during the anaphase stage of the mitotic cell division. During the anaphase stage of the mitotic cell division, because of the splitting of the centromere, here the segregation of homologous chromosomes does not take place. Instead of the segregation of homologous chromosomes, only the separation of sister chromatids takes place. Is it clear? Because of the splitting of the centromere, here segregation of homologous chromosomes does not take place during the anaphase stage of the mitotic cell division. Instead of the segregation of homologous chromosomes, here only the separation of sister chromatids takes place. So let me write it here. Because of the splitting of centromere segregation of homologous chromosomes does not take place, and instead separation of sister chromatid occurs. Is it clear? And here we need to understand that when the specific sister chromatid, when it successfully migrates towards the opposite spindle pole, initially the sister chromatid, it takes the advantage of the splitted centromere. So here, by taking the advantage of the splitted centromere, the sister chromatid, it is ready to migrate towards the opposite spindle. Is it clear? So here, this is regarding the anaphase stage of the mitotic cell division. and then after we will be studying regarding the telophase stage of the mitotic cell division in detail. So let me arrange the things here. And when the mitotic cell division, when it successfully progresses from the anaphase stage to the telophase stage, so here during the telophase stage, successful migration of the sister chromatids towards the spindle pole it takes place. So here after the successful migration of the sister chromatids towards the opposite spindle pole, now here the specific sister chromatids they lose their individuality. Is it clear? When the specific sister chromatids, when they successfully migrate towards the opposite spindle pole, now here the specific sister chromatids they lose their individuality and the specific chromosomes they are no more they are visible under microscopic observations. Is it clear? During the telophase stage, when the sister chromatids migrate towards the opposite spindle pole, the specific chromosomes lose their individuality, right? And here chromosomes they are no more visible under microscopic observations. And here the various subcellular organelles will successfully disappear during the late prophase. Again, all the subcellular organelles like nucleolus, endoplasmic reticulum, Golgi complex, again they reappear during the telophase stage. Is it clear? During the telophase stage, all the subcellular organelles like nucleus, nuclear envelope, nucleolus, endoplasmic reticulum, Golgi complex reappear, right? So here at the end of the telophase stage, from one single parental cell, two daughter cells are obtained. And here the specific daughter cell, likewise the parental cell, even the specific daughter cells they has same chromosomal number. So the reason why the mitotic cell division, here it is otherwise known as equational division. Is it clear? Likewise the parental cell, even the two daughter cells, they possess similar chromosomal number. So the reason why mitotic cell division, or it is otherwise known as equational division. And then after we will be studying regarding cytokinesis. So here what is cytokinesis? So let me arrange the things here. And then after we will be studying regarding cytokinesis. So here what is cytokinesis? So mitotic cell division, it is not only responsible in regulating the production of two daughter cells and the maintenance of the constant chromosomal number, and even in order to regulate the production of two daughter cells, the mitotic cell division, it takes the advantage of a specific mechanism termed as cytokinesis. So as already mentioned, mitotic cell division in general, it begins with division of nucleus, which is termed as karyokinesis, and it ends with division of cytoplasm, which is termed as cytokinesis. Is it clear? So let me write it here. Division of cytoplasm is termed as cytokinesis. Cytokinesis, right? So here cytokinesis mechanism, it is observed both in case of the animal cell and even in case of the plant cell. But here the cytokinesis mechanism which occurs in case of the plant cell, it remains significantly vary from the cytokinesis mechanism which occur in case of the animal cell. Is it clear? So initially we will be studying regarding the cytokinesis mechanism which occur in case of the animal cell. So here in case of animal cell, in order to regulate the cytokinesis mechanism, here there is a requirement of a specific cleavage furrow. So here appearance of cleavage furrow, it successfully takes place within the plasma membrane of the animal cell. Is it clear? So let me write it here. In order to regulate cytokinesis, cleavage furrow is required, right? Cleavage furrow is required. So here appearance of cleavage furrow, it takes place within the plasma membrane of the animal cell. So initially the specific cleavage furrow centripetally, that means from the exterior side towards the interior side, it successfully proceeds. So here once when the cleavage furrow, it undergoes deepening, now here the specific cleavage furrow, it successfully joins at the center and it is responsible for the division of the cytoplasm into two dot cells. Is it clear? The specific cleavage furrow centripetally it migrates from the exterior side towards the interior side. So here once when the specific cleavage furrow, when it joins at the center, it is responsible for the division of the cytoplasm into two dot cells. Is it clear? Deepening of the cleavage furrow and it is responsible for the division of the cytoplasm into two dot cells. In addition to this, the cytokinesis mechanism which occurs in case of the plant cell, it remains significantly vary from the cytokinesis mechanism which occur in case of the animal cell. So here in case of the plant cell, in addition to the cell membrane, even there is an availability of one more other specific layer termed as cell wall. So here in order to regulate the formation of cell wall, here there is a requirement of a specific structure termed as early cell plate structure. Is it clear? So let me write it here. In case of plant cell, in order to regulate cytokinesis, formation of early cell plate is required. So here, how the formation of early cell plate it takes place in case of the plant cell. So here in order to understand this particular concept, here we need to take the advantage of the concern diagram. So initially in order to regulate the formation of early cell plate, here there is a requirement of a specialized structure termed as phragmoplast. So here what is phragmoplast here? Phragmoplast, it is an association of microtubules and actin filaments. So here once when the microtubules and actin filaments, once when they aggregate together, it leads to the formation of a specific structure termed as phragmoplast. Is it clear? So let me write it here. An array of microtubules and actin filaments form phragmoplast. Is it clear? So here microtubules in association with actin filaments, it lead to the formation of phragmoplast. And to the concerned phragmoplast, as already mentioned, at the end of the telophase, the various subcellular organelles like endoplasmic reticulum, Golgi complex and nucleolus, they reappear. So here, in case of plant cell, Golgi apparatus, it is responsible for regulating the carbohydrate metabolism. So here, one of the specific carbohydrate like cellulose, it successfully synthesizes within the Golgi apparatus. So here, after the successful production of the specific cellulose within the Golgi apparatus, now here the specific cellulose, it successfully packs within the Golgi derived secretory vesicle. Is it clear? After the successful production of cellulose within the Golgi apparatus, now here the specific cellulose in the means of cell wall precursor, it successfully packs within the Golgi derived secretory vesicle. So let me show it here. So here it is nothing but Golgi derived secretory vesicle. Golgi derived secretory vesicle, right? Golgi derived secretory vesicle. So within the Golgi derived secretory vesicle, here there is an availability of cell wall precursor. Is it clear? Cell wall precursor. And here the cell wall precursor, here it is nothing but cellulose. The specific cellulose which successfully synthesizes within the Golgi apparatus, it successfully packs within the Golgi derived secretory vesicle. And the Golgi derived secretory vesicle, it successfully packs or it successfully integrates with the phragmoplast. Fusion of the Golgi derived secretory vesicle with the phragmoplast, it is responsible for the formation of early cell plate. Is it clear? So here this is the specific cytokinesis mechanism which occurs in case of the plant cell. And in addition to this, here we need to understand that in order to regulate the production of, or here in order to regulate the distribution of the various subcellular organelles like plastids and even the mitochondria to the specific plant cell, here cytokinesis mechanism, it plays an important role. Is it clear? So let me write it here. In plant cell, during cytokinesis, distribution of plastids and mitochondria to the daughter cell takes place. Right? So here this is regarding mitosis and its regulation from cell cycle and cell division in detail. And in the next upcoming lecture session, we will be studying regarding meiosis in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.